Assalamu alaikum everyone. Welcome back to a new session in online physiology, and today we will discuss the B lymphocytes as the source of antibody-mediated immunity. We discussed the T lymphocytes last time, and we will follow the same algorithm for B lymphocytes as well. So to begin with, like all other cells in circulation, all the blood cells in circulation, the B lymphocytes too are produced in the bone marrow, because the lymphoid stem cells which gave birth to the B lymphocytes reside in the bone marrow. However, soon after birth, while they are still immature, they migrate to their pre-processing sites. Now here, unlike the T lymphocytes, which are pre-processed in the thymus, the B lymphocytes are pre-processed in the liver and bone marrow. Pre-processing in the liver occurs during the mid-fetal life. However, in late fetal life and after birth, their pre-processing shifts to the bone marrow. Okay. Now, during the pre-processing, they not only proliferate to increase their numbers, but also they differentiate into more and more mature cells capable of secreting antibodies. Okay. Now here it is worth mentioning that in terms of diversity, the B lymphocytes exhibit far greater diversity than the T lymphocytes in terms of antigen specificity and reactivity, so that we have tens of millions of B lymphocytes having specific reactivities against specific antigens. After their pre-processing at these sites, they migrate to their storage sites. And as we discussed for T lymphocytes, they are stored in the lymphoid tissue, most extensively in the lymph nodes. But this is not limited to the lymph nodes. They are also stored in special lymphoid tissue elsewhere in the body, like the spleen, the thymus, the gastrointestinal tract, and the bone marrow itself, where they keep on waiting for possible encounter with the antigen against which they show specific reactivity. Okay? Still later, if we talk about their activation, they are a bit privileged as compared to T lymphocytes. The T lymphocytes are entirely dependent on other cells, the antigen presenting cells, for their activation. The B lymphocytes, however, can be stimulated directly by the antigen. Okay. However, that in terms of magnitude, that direct stimulation is of low magnitude, 1+. Okay. And here I wish to mention that the B lymphocytes themselves are the antigen presenting cells. The antigen can bind with the B cell receptors, with the receptors on their surface, and cause their activation. Okay, and the B lymphocytes can offer those pre-processed antigens to other cells. Okay, and secondly, once the B lymphocytes have a direct encounter with the antigens they are reactive to, they offer those antigens to the helper T cells, and the helper T cells in turn secrete those lymphokines which activate the B lymphocytes to a far greater magnitude. Okay, so that we have, in terms of magnitude of activation, that indirect route of activation aids or facilitates their further activation. And once they are activated, what they do? They proliferate extensively to increase their numbers. They not only transform into what we call plasma cells. Not every B lymphocyte is a plasma cell. We call them plasma cells only when they have been activated by a certain antigen against which they are reactive, and they transform and change their character and are capable of secreting antibodies only when they become plasma cells. Okay? The plasma cells secrete antibodies to fight against the invading microbe or the culprit antigen. But at the same time, that progeny of B lymphocytes that is activated by a certain antigen gives rise to what we call memory B cells. Those memory B cells repopulate the bone marrow. They stay dormant there till re-exposure to the same antigen so that they can mount a rapid, quicker and a more potent response in terms of antibody production. Okay, now at this point I would like to educate you a few things about the antibodies. Now each antibody is composed of heavy chains shown here in black and light chains shown here in blue. Okay, even though the number of heavy and light chains do vary between different antibodies, but most of them have the light chains, 2 light chains and 2 heavy chains. Okay, now here the heavy chains are bound together by disulfide bonds. Similarly, the light chains are bound with the heavy chains through sulfide linkages. Okay, this is what keeps them together. And here we have 2 parts of each antibody. This is the proximal part and this is the distal part. They are bound to the cell surface, on the surface of the B lymphocytes, through their heavy parts, through the heavy chains. Okay, and they bind to the antigen at their more distal parts. Here distally, only the distal part, say 1/2, the distal 1/4 of the heavy chain and the distal 1/2 of the light chain constitute what we call the variable portion. Okay, while the more proximal part, the remaining part, constitutes what we call from here till here, what we call the constant portion. Okay, if you want to make it sound difficult, we also call the variable portion the fab portion. We also call the constant portion the FC portion. Okay, now depending upon their properties, the variable portion, also called the fab portion, determines antigen specificity. Only a certain antigen can bind at this point. Okay, and this is all because we have a unique structural organization of amino acids in the variable portion that fits only with a certain specific antigen. So that portion, the variable portion, determines specificity of the antigen it will react with, while the constant portion determines other biochemical and biological properties of the antibody, like it could be the diffusing potential of the antibody in different tissues, its adherence to various structure in different tissues, and its ability to activate complement, and so on. Okay, now here, for example, if this is the biological portion, it can activate a certain antigen. Say we have the surround antigen that can bind with the variable portion. Okay, and this second arm too will bind a similar antigen. If we could presume for a moment that this limb, this variable portion, belongs to some other antibody, so that in that case it will be able to bind a different antigen, not this one. Okay, so this is all about the general structure of the antibodies, the variable portion and the constant portion. Now we move on to discuss the mechanisms of action of different antibodies. Well, the antibodies act against the antigens are invading microbes in 2 ways. They either act directly on their own against the antigen, or they may act indirectly through activation of some other component of the immune system. So to begin with, the most common mechanisms of action of antibodies include agglutination, precipitation, neutralization, and lysis. Here we need to discuss them in bit greater detail, beginning with the agglutination. If these are the multiple antigens, and there are so many of them, the antibodies bind these antigens, say this one here, this one here, this one here, and this is how. So we have multiple antigens here in this case, and they are bound together by multiple antibodies so as to make a clump. They are clumped together, and clumping of different antigens helps in two ways. First, clumping together decreases their mobility, okay, and they can't move that freely. Secondly, by definition, most of the times an antigen for a substance to be antigenic, that is to be capable of causing disease, it needs to have a molecular weight of greater than 8,000. Okay, so if we have multiple small particles, multiple ones, so that each one of them doesn't qualify to be an antigen because they have a molecular weight less than 8,000. So clumping together through antibodies can make them qualify an antigen and cause arousal of the immune system. Okay. Secondly, if they have been clumped together this way, their mobility decreases significantly, and then they can be taken up by the phagocytic cells like macrophages, etc., etc. Okay. Still later, we have precipitation. Some antigens remain soluble. They are soluble, like the tetanus toxoid is one such antigen that is soluble in the plasma. If the antibody combines with this antigen, the antigen-antibody complex becomes insoluble. So owing to its insolubility, it precipitates in the plasma. That not only makes it more visible, but also prone to phagocytosis by the immune system or action of the immune system in some other way. Okay. Still later, we have neutralization. So for example, if this is one antigen, okay, 8 times the whole antigen or the whole microbe that invades the tissues is not that dangerous. It has only a certain part of it that has a unique sequence of molecular groups that makes it dangerous. For example, this whole microbe or whole antigen may not be problematic. Rather, it's only certain portion of it, say this one, may carry a certain sequence of amino acids or other molecules to cause damage to the body. So what antibodies do, they just mask the antigenic site on the microbe or the whole antigen. Okay, this is like if we can take care of someone owing to physical proportions, okay, and that fellow or that opponent comes up with a knife or a pistol in his hand, so the balance of power tilts in his favor. Okay. Similarly, the whole microbe or the whole antigen may not be problematic. Only a certain part of it may cause disease, and the antibodies will simply mask that dangerous part. Okay. Like we take hold of his hand with the knife or with the pistol. Okay. Still later, sometimes the antibodies can directly attack the invading microbe or the antigen and punch out holes, like say this one. It may punch out holes in its cell membrane, thereby directly cause killing of the invading microbe. Direct lysis through attack on its cell membrane. Okay. So, however, I need to mention, killing by the direct route is not an efficient way of action of the antibodies. Most of the time, most potent action comes from indirect mechanisms of action, and those indirect actions include activation of the complement. Certain antibodies, not all of them, for example, only the IgG, IgM, and now there is some evidence that IgA can cause activation of the complement. That is a system of a system of over 20 plasma proteins that can aid the immune system contain antigens and microbes in different ways that facilitate the immune response. So the IgM, IgG, and to some extent IgA cause activation of the complement system so as to facilitate the immune system in containing the threat, that particular antigen or disease. We will discuss the complement in bit more detail in a dedicated lecture, but for the time being it should suffice that antibodies work either directly through agglutination, precipitation, neutralization, or lysis, or indirectly through activation of the complement system. Okay, at undergraduate level, this much detail should suffice for most of you. However, there are some confusing titles, some confusing parts of the discussion which we will cover in a dedicated lecture that is coming shortly. Thank you.